tremendous progress in the synthesis of inorganic nanoparticles with controlled morphology, size, and surface properties. And with these developments, has raised the perspective to transform these materials into multifunctional nanoplatforms for thermagnostic applications. So indeed, there is a possibility to combine their inherent imaging property that can be of different kind, depending on the composition of the nanomaterials, with the conjugation at the surface of the nanoparticle of, for example, targeting ligands. And here you can see some example of cancer-related biomarkers that could be targeted. And also the possibility to introduce at the surface of these nanoparticles therapeutic cargoes that can be released upon the action of endogenous or external stimuli. And finally, there is also the possibility to combine an additional imaging probe at the surface of these nanomaterials in order to overcome the limitation of a unique imaging modality. So in our laboratory, we have been interested in the development of harmonic nanoparticles for different types of applications. So these materials, as you can see, they are based on non-centrosymmetric crystal structures, and such as here bismuth ferrite and also lithium niobate, which are our favored nanomaterials for the different applications that I will share with you. And these nanoparticles are really characterized by their second harmonic scattering properties. So meaning that, as you can see here, when two photons coinciding in time and space are combined by the system, while in fact their frequencies are not adding up to an existing transition of the material, there is a possibility to generate an emitting photon at exactly twice the frequency and half the wavelength. So in this respect, they differ from upconversion nanoparticles that are represented here, where in fact they absorb two photons. And these photons here add their frequencies to match the difference between two molecular levels. So in the harmonic generation process, there is no energy which is deposited on the sample, because here you only reach a virtual level. And on top of that, the process can be generated at any incoming wavelengths generated by an ultra-fast laser pulsed system. So the advantages of harmonic nanoparticles rely on several aspects. The first one is the tunability of the excitation wavelengths that you can really adjust in a region where the absorption on the scattering of the analyzed medium is minimal. So here you can see in red all the excitation wavelengths that are accessible, and in blue you see the harmonic emission that you can retrieve. Also, in addition, they benefit for a long-term photostability in comparison with fluorescent labels, as shown here in this cell culture, which has been incubated with harmonic nanoparticles. So you can see here that the emission of the nanoparticles is highly stable over time, even after several hours of excitation, while in fact the signal from the fluorescent dye is vanishing after a few minutes of excitation. So these are two advantages of those nanomaterials. And in addition, they also benefit from quite narrow emission bands and signals, and also a high spatial resolution. So the advantage that are shown here are not only limited to the second generation harmonic process, but in imaging setups, you can record also the third harmonic generation process, meaning that not only two photons can be combined, giving back one emitting photon, three photons can be combined, giving back one emitting photon at a third of the wavelength and 3 times the frequency. So this is quite advantageous when you want to make imaging in quite complex environment. So for example, here you have the multi-harmonic emission of bare bismuth-ferrite nanoparticles, where you can see both the second and the third harmonic signal. And here, the image colocalizing these two emissions. So this colocalization process can be used, for example, if you want to make imaging in tissues. So here we have an example of an excised cancer tissue. And here is just the image of the tissue without any labeling. And as you can see, there is a strong endogenous second and third harmonic signals which come from the collagen and the lipid component of these tumor tissues. And these constituents are very abundant in cancer tissues. After incubation with bismuth-ferrite harmonic nanoparticles and application of the colocalization procedure, you can see that there is a clear and very selective identification of the nanoparticles, which are represented here by the white spot, thanks to the background rejection, because as you can see here, in the semi-log histogram of pixel intensity at both the second and the third harmonic, we have covered the whole detection range. And it highlights the discrimination of the nanoparticles, so the harmonic nanoparticles, like you can see here in blue, which are the only element that colocalize the second and the third harmonic emission signals. So this protocol allows to further investigate the use of harmonic nanoparticles in cell tracking. And for example, stem cells have the potential to be developed as therapeutic products for the treatment of several diseases, such as here, the Duchenne muscular dystrophy, which is a fatal X-linked recessive muscle disease, which results in progressive muscle degeneration with fatal issues at the age of around 20 to 30 years old. So in order to assess the potential of human muscle stem cells, which are called HMU stem cells, for the treatment of this disease, it is really important to address, among other questions, their biodistribution and their engraftment properties. So to do that, we have imagined that these harmonic nanoparticles appear as excellent candidates for tracking those cells after engraftment and after injection. And so what we did, we made first the covering of those nanoparticles by PEG derivative, and then we incubated HMU stem cells with those nanoparticles at a concentration of about 12.5 microgram per milliliter of solution. And the excitation both at 1040 nanometer and 1003 nanometer allowed to image the harmonic nanoparticle in all channels, as you can see here, and also to really clearly distinguish those nanoparticles from the background signals from the cells, which have been at the same time stained with the phalloidine Alexa 555 dye. So then what we analyze is how deep these nanoparticles could go in tissue. And for this, we incubated tissues with those nanoparticles, and we retrieve the images of those tissues after 12 hours of incubation. So here you have the full image with the excitation at 1300 nanometer, where you can see the nanoparticles, which in some area of the tissue form small aggregates. And what is interesting is that to get a better resolution and distinction of those nanoparticles, it is possible to apply the colocalization protocols that we have discussed before. And here you can see that the nanoparticles are clearly present up to more than 1 millimeter depth in the tissue. So based on these first result, we decided then to inject, in fact, HMU stem cells labeled with harmonic nanoparticles that we have characterized also here to see how the nanoparticles are internalized by those cells. And so, in fact, we injected these nanoparticles in an animal model, so it was in mice. And then we retrieved the tissue 24 hours post injection, and we analyze the muscle tissues. And so, as you can see, ex vivo analysis of this tissue showed either here and also in this second image we see here, that the nanoparticles, which are these blue spots, exclusively colocalized with the fluorescent staining the cells of interest, which were the ones which were injected. And you clearly see that the nanoparticles are not present in other areas of those tissues. So that you can really track those cells thanks to the presence of this harmonic nanoparticle over a long time. And what we analyze also is that in cell culture, so in vitro, even 14 days after culture, and after, in fact, multiplication of those cells, you still have nanoparticles which are present and visible in those cells. So after these first applications, which relate to nanoparticles which are just simply covered by PEG derivative, we can move to surface functionalization of those nanomaterials to reach other applications, such as, for example, targeted cancer imaging. So in our group, we envisage the decoration of pegylated harmonic nanoparticles with targeting ligand addressing cancer cells and tumor microenvironments. So here you see selected example of relevant biomarkers for cancer bioimaging. So for example, alpha-V beta-3 integrin, which is involved in cell adhesion, migration, and proliferation. It's targeted by, in fact, those cyclic peptides represented here. So what we did first is to make a modification of this peptide in order to introduce a spacer that will be further used to make the conjugation at the surface of the nanoparticles. We then analyzed in different cancer models, and here you have the example of a lung cancer model, that the tumoral cells are associated with these nanoparticles, as we can see here with these brown spots, while non-tumoral cells are almost non-associated with the same functionalized nanoparticles. Then we choose another biomarker, which is fibroblast activation protein alpha, which is a prolyl-specific protease, selectively expressed generally in the microenvironment of the tumor because it's expressed by reactive stromal fibroblasts. This protein is involved in carcinogenesis and is generally targeted by selective inhibitor mimicking a GLI-PRO sequence. So previous work by the group of Jensen has showed that this specific substituent on a GLI-PRO sequence is really useful in order to generate an inhibitor which is selective toward fibroblast activation protein alpha and which only shows a very light, let's say, inhibition of other related enzymes which have a close structure to FAP alpha. So in our group, based on this work, we developed an analog of this substrate, which is linked here to a small spacer that will be then used to make the conjugation at the surface of the nanoparticles. And we observed that this ligand also has a very specific recognition of the human recombinant enzyme and has almost no activity on the close enzyme to this to FAP alpha. Then comes the conjugation of those ligands at the surface of the nanoparticles. And in fact, we based our protocol on the possibility to make a click reaction at the surface of the nanoparticle. And this click reaction is sought by using azido group that are introduced on the coating polymer and an activated triple bond, which is represented here, and that we introduce on the targeting ligand in order to make a spontaneous here 3+2 cycloaddition, which give rise to the covalent linkage of the nanoparticles to the targeting ligand without the need for any additional cross-linker or chemical reagent. So here you have an example of the conjugation that was performed using the specific inhibitor of fibroblast activation protein alpha. So we followed and we monitored the different steps of the functionalization by measuring the mean hydrodynamic diameter of those nanoparticles and their zeta potential. And you see that here the values are consistent with first the introduction of amino group at the surface of the nanoparticle. So we go from a negatively charged surface to a positively charged surface. And then we have the introduction of the ligand, which does not affect the zeta potential of the nanoparticles. And by functionalization, we also increased the dispersity and the dispersity of these nanoparticles, which had the tendency to aggregate when they were not coated and functionalized. So here we have the stem analysis of those nanoparticles, and we see that the coating and functionalization layers is around 8 nanometers. Then we used these functionalized nanoparticles for making targeted cancer cell imaging. And here you have an example of different lung carcinoma cells, which were efficiently labeled and selectively labeled with nanoparticles. And here you can see the emission of these nanoparticles when we add an excitation of the seed stem at 790 nanometer. Then another important aspect that we investigated with these nanoparticles was the possibility to combine the multi-harmonic imaging capabilities, so using both the second and the third harmonic generation, with magnetic resonance imaging, thanks to the conjugation at the surface of the nanoparticle of gadolinium-based chelates. So here we have an example of a compound that was described in the literature to be a very good chelating agent for gadolinium. And so we decided to make a modification of this initial ligand in order to be able to make the conjugation at the surface of the nanoparticle. So we developed a conversion synthesis of this new ligand, which allows through this core in fact, the complexation of the gadolinium ion, and through this tail, in fact, the conjugation at the surface of the nanoparticles. So from this intermediate ligand, upon saponification condition, you can get to the chelation of gadolinium ion, and then by a click reaction at the surface of lithium niobate coated nanoparticle, you end up with these conjugates, which combine the harmonic nanoparticle core with a gadolinium chelate. So we also followed the conjugation through the characterization of the different steps of these nanoparticle functionalization. And we first verified that these modified nanoparticles maintained their ability to make multiphoton imaging in, for example, cancer cell culture. This was the case here for prostate cancer cells, where you can see that you can get both the second and the third harmonic signal of the nanoparticles which are labeling those cells. Then we went to the analysis of the gadolinium done by XPS, where you can clearly see the signal coming from gadolinium, while the non-functionalized nanoparticles have no emitting signal in XPS. Then in term analysis, it's also very clear that you have the elements that correspond to the composition of the material. So for example, niobium and oxygen. Then you have the silica layer, which is around the nanoparticles. And finally, here you can see the gadolinium distribution at the surface of the nanoparticles. So we have a covalent conjugation of those gadolinium chelate on the coated surface of the nanoparticles. Then we analyze the relaxation time and rate of those nanoparticles, and you can see here that you have an increase of the relaxation rates of water proton, both in longitudinal and transverse measurements at 30 and 60 megahertz, while the non-functionalized nanoparticle has no effect. Then, after calculation of the reactivities, you can see that with the functionalized nanoparticles, we are in the range of the contrast agent, the commercial contrast agent based on gadolinium chelates. So then we went to further analysis of those nanomaterials for MRI imaging. At 9.4 Tesla, we analyzed the T1 spin lattice relaxation times using, in fact, a phantom that was based on an agarose gel using 6 different concentrations of those nanoparticles in the suspension. And here you can see the clear dependence on of the T1 spin lattice relaxation time that is represented here on the concentration of the nanoparticles in the suspension. Then we move to MRI for the quantitative T1 mapping. And here you can see, this is the image of the phantom, that there is a strong contrast enhancement, which is correlated directly with the concentration of the nanoparticles in the sample. So this combined system really opens the way for multimodal imaging using those harmonic nanoparticles. Finally, the last aspect that we explored was the possibility to use the harmonic signal that is generated by the nanoparticles, in fact, to trigger the release of caged molecular cargoes that will be introduced at the surface of the nanoparticles, giving the possibility for decoupled imaging and photoactivation protocols. So this was previously reported, for example, for up-conversion nanoparticles, but these have been never developed for harmonic nanoparticles before. So most of the photosensitive spacers, they are activated by UVV's excitation. And we know that this UVV's excitation, as low tissue penetration depths, can be toxic for cell and tissues and also promotes photoreaction. So there is a high advantage to use near-infrared excitation, which has a deeper tissue penetration, also a minimal absorption and scattering of tissues in this window, and also reduced toxicity. So what we performed is to introduce a coumarinil-based spacer between a molecular cargo and the surface of the nanoparticles, and then to try to release the cargo using the harmonic emission of the nanoparticles. So we ended up with the synthesis of this ligand, which contained the coumarinil spacer. Here you have tryptophan as a model molecular cargo, and the spacer allowing the conjugation at the surface of the nanoparticles through this click reaction. So then we irradiated the nanoparticle at 790 nanometer, and we observed is the harmonic emission was able to trigger the release of tryptophan, which was quantified by AZMS using UHPLC, so ultra high performance liquid chromatography. And as you can see here, the cumulative release of tryptophan over time. So already after 15 minutes of excitation, you reach more than 60% release of the molecular cargo. And after 30 minutes, you are between 70 and 80% release of the molecular cargo. And by measuring the kinetic of this release, you can see that this is really due to the harmonic emission of the nanoparticles, as the ligand itself has only a very small release due to the two absorption capacity of, in fact, the coumarin spacer. So this was really the first example of a photo-controlled release as a result of the harmonic emission of harmonic nanoparticles. And we were able then to apply this process to the release of anti-cancer drug. And one of our targets was to use an inhibitor of the epithelial growth factor receptor tyrosine kinase domain, which is EGFR, because this receptor is overexpressed and sometimes mutated in a variety of tumors. And we know that erlotinib, which is a selective inhibitor of the tyrosine kinase domain of EGFR, is an FDA-approved anti-cancer drug with limited side effects. So here you see the structure of the commercial erlotinib. And then we decided to go for the modification of this drug in order to introduce this spacer that will give us the opportunity to make the functionalization at the surface of the nanoparticle. So we developed a synthesis toward this analog, and then we conjugated this analog to, in fact, the coumarin spacer, and finally to the activated triple bond, which gives the possibility to make the click reaction at the surface of the nanoparticles. So these are the nanoparticles that we obtained, which were characterized first by TEM, where you can see, in fact, the component of the inorganic core, the silica shell, and also then the functionalization. We see also the evolution of the diameter and the zeta potential during the different steps of the protocols. And then we went for the imaging properties and drug release properties of the nanoparticles. So first, by irradiation at 790 nanometer, what you can see is that you can retrieve the second harmonic signal of the nanoparticles as well as the fluorescent signal of coumarin, which is an additional proof that we have made the conjugation of the ligand at the surface of the nanoparticles. Then we also image those nanoparticles at a higher wavelength in order not to trigger first the release of the molecular cargo, but just to see if we can make the imaging, for example, of prostate cancer cells, as you can see here on those pictures, by retrieving the third harmonic signal and the second harmonic signal, which are colocalized. Then we evaluated the cytotoxicity of these nanoparticles, and you can see that in average, at different concentration, the nanoparticles themselves are quite close to the negative control, which is thus the cells toward, in fact, ethanol. Then we went to the controlled release, but first to do that, we had to verify that the erlotinib derivative is able to induce the same inhibition as the commercial drug. And whereas after 24 hours, this was not observed, but after 72 hours of exposure, you can see that erlotinib, so the commercial drug, and our derivative result in quite similar activity toward, in fact, the inhibition of cell growth. So then we moved on with the control release by irradiation of the conjugated nanoparticles at 790 nanometer, and we measured the variability of the cells 72 hours post-irradiation. And the irradiation time was kept at 15 minutes. And so here you can see the optical density showing the variability of the cells without any addition of nanoparticle, just the irradiation. Here you can see the cells in the presence of the coated nanoparticles, so the ones which do not have the erlotinib at the surface. And then here you can see the cells with the functionalized nanoparticle and the irradiation, and the cells with the inhibitor itself without the nanoparticles. And you can see that the inhibition of cell growth is similar to the drug itself. So this was very encouraging results. And now we are combining at the surface of the nanoparticles these molecular cargoes with also the possibility of adding a gadolinium chelate so that you have a multimodal nanoparticles allowing dual imaging plus drug release. So I hope that I have shown you some nice application of those nanomaterials, which are really interesting, properties. So you can make cell imaging by using the near infrared window for the excitation. Thanks to the combination of the multi-harmonic emission of those nanoparticles, you can make imaging in very complex tissues. You can also track cells in those complex tissues. You can make controlled drug release. So I showed you the example of erlotinib, but we also perform in the lab the release of other anti-cancer drugs, such as chlorambucil. And finally, you can also move completely the excitation wavelengths to the region of the visible light. And by this means, those nanoparticles will emit in the deep UV region, and this will directly trigger cell photo damage. So this is an application that I didn't show you, but that we also investigated in the course of these studies. So finally, I would like to thank my team who worked on this project. So the team members are, you can see them here. They all did a PhD in my group in relation to that subject. I would like to thank also the very nice collaboration that we have with the University of Geneva for the imaging platform. Our partners in France for the in vivo cell tracking activities, the SIM laboratory who is producing the bare nanomaterials, so the inorganic core structure, and the University of Lausanne with who we are collaborating for the cancer cell experiments. And finally, our sponsors who have funded all this research.